morning, everyone. On behalf of the student chapter of the Eastern Pennsylvania branch of ASM, I would like to welcome all of you to the 29th Annual Philadelphia Infection and Immunity Forum. My name is Lauren Springer, and I'm a third-year PhD student at Thomas Jefferson University. I'm co-president with Natalia Benavides of the student chapter. I would like to start off by thanking all of you for being here bright and early on a Friday morning. This year is a little different than our usual forum, as COVID-19 has led us to hold this event virtually for the first time. Nonetheless, we are very excited to be here with all of you today. Before we get started with our exciting lineup of talks, I would like to take a moment to thank our generous sponsors whose financial contributions have made today's event possible. First, we would like to thank PBL-ASM Science, our annual sponsor of the Sydney Pesca Lecture, which will be our last talk of the day in the afternoon. We would also like to thank ASM National for providing us with a conference grant. Finally, I would like to thank the incredible EPA-ASM student chapter team for working tirelessly for the past year in organizing this event. Our amazing faculty advisor, Dr. Michelle Kutzler, for mentoring us and answering our countless questions at every step of the way. The student officers, my co-president, Natalia Benavides, our co-vice presidents, Matthew Bell and Kayla Sakaras, Secretary Ronald Lucarelli, and Treasurer Marissa Egan. Thank you to all of you for stepping up to the challenge of organizing today's forum. In addition to today's forum, we also organized the May monthly meeting during which poster award winners from previous year's Infection Immunity Forum give an oral presentation of their research, along with one invited faculty speaker selected by student officers. This year, we had the pleasure of hosting Dr. Matthias Schnell. We also organized a social event for our student chapters, and for the past few years, we've been going to the Franklin Institute's Science After Hours. We also collaborate with the Education Subcommittee to provide opportunities for professional development. Unfortunately, our event planned for spring 2020 had to be canceled given the current circumstances, but we hope to work with them again in the future so our chapter members can receive training and judge the George Washington Carver Science Fair. We highly encourage students to join the chapter. It's free and it helps us receive funding to hold more events like the ones I've shared with you today. Also, it's really easy to sign up. You visit EPA-ASM.org here, and this will lead you to our main homepage, where there will be the link Join Today. Click on this link to register. This will lead you to our main homepage, where there will be the link Join Today. Oh, sorry. And with that, I would like to thank you again for attending today's forum and turn over to Dr. Dieter Schifferle, the president of the Eastern PA branch of Pennsylvania.
Okay. So welcome all again. I'd like again to quickly thank the leadership of the student chapter of our branch, which just presented itself, as well as Dr. Michelle Kutzler, our president-elect. They all have worked extremely hard to organize this Zoom meeting, and as well as Dr. Vincent Tam, our skilled video, Zoom, YouTube jockey. I also would like now to take this occasion to remind everybody, maybe next slide, that our EPA-ASM site also includes our list of monthly seminars. So 10 days ago, we had Dan Portnoy, and then in January, we're gonna have Dr. John McAlanas from Harvard, in February, Isabel Coppens, etc. You can see the whole list there. So we're a very active branch of the ASM, and I hope you all will be able to make it. Now, next slide, please. If you miss any seminar, you can also find these seminars on YouTube links whenever the speaker allows us to put these talks on YouTube. So you can find actually these talks under our website under archives, as shown at the bottom of these slides. Next slide. Since we're a branch of the American Society of Microbiology, I'd like also to advertise National ASM. First, by showing the link at the bottom there, where you can become a member to find information on the community of microbiologists, local to international, create networks for colleagues, and also information for students and postdocs, professional development, and finally information on scientific meetings, webinars, etc. Particularly, next slide, please. The ASM Microbe Online, which has many seminars on SARS-CoV-2 and COVID-19. If you haven't read enough of it by now. You can go there and hear many very interesting seminars, as well as other scientific presentations such as e-posters, etc. Next slide. Finally, I have some logistical details for the event. For this online forum style, attendees will be muted so that each talk can go to completion. Please, please write your question for the presenters in the chat. Questions will be answered during the allocated time at the end of each talk. If there is not enough time to answer all questions, having them written in the chat will allow the speakers to answer them later with you. With this in consideration, speakers, please be mindful of presentation time. We have lots of amazing speakers at the event today, and we would really like to have them all take a full advantage of their time and for this event to be completed in a timely manner. Finally, lunch will be from 11:15 a.m. to 1:15 p.m., during which we'll have posters presentations. We have two sessions. Session 1 will be for the first hour from 11:15 to 12:15. There, all the odd number posters will be presented, and session 2 will have the even poster presentations from 12:15 to 1:15 p.m. Okay, so now I leave Natalia Benavides, the student chapter co-president, to introduce our first speaker, Dr. Watnick.
Thank you.
Lauren, here is Natalia. Very good.
So for our first speaker today, I would like to introduce Dr. Paula Watnick. Dr. Watnick is a senior associate physician in pediatrics of the Division of Infectious Disease at Boston Children's Hospital. She's also an associate professor of pediatrics at Harvard Medical School. She received her PhD from the California Institute of Technology and her MD from Yale University. Over the past 10 years, Dr. Watnick and her laboratory have used bacterial genetics, genomics, proteomics, and metabolomic approaches to probe the interaction of microbes with the host intestine. Specifically, her lab is interested in understanding the impact of intestinal pathogens on host metabolism and nutrition, the development of biofilm-based antigen and enzyme delivery platforms, as well as discovering novel natural products with antimicrobial activity. Today, we have the wonderful pleasure of listening to her talk titled How Microbial Metabolites Control Innate Immunity and Metabolism in a Model Host Intestine. Please welcome Dr. Paula Watnick.
So thank you very much. I'm just gonna, let me see if I can share screen.
I will hand it over to Matt Bell, who's our co-VP president or co-vice president of EPA-ASM student chapter.
Thanks, Natalia. So like Natalia said, I'm Matt Bell from Drexel University, and along with Kayla, I'm co-vice president of the student chapter. And today I would like to introduce our next speaker and welcome him, Jean-Bernard Lubin. He is a postdoctoral fellow in a lab of Dr. Michael Silverman at the Children's Hospital of Philadelphia. And he will be presenting his talk titled The Gnotobiotic Mouse Model of Pediatric Post-Commensal Interactions.
Hello? Can everyone hear me okay now? Okay,
Yep,
thank you.
We can hear you now.
All right. Okay, is my title screen up? Sorry, I can't tell.
Yeah, we can see it.
Okay, thank you. All right. Thank you and good morning, everyone. I'll be going over my talk, which is titled A Gnotobiotic Mouse Model Pediatric Host Commensal Interactions, and share some of the data I've gathered developing this model. I'll start us off with some basic introduction to the microbiome, and particularly my point of focus of the microbiome is actually the pediatric pre-weaning microbiome, which as of this point has been well characterized to be unique in function and composition to that of a post-weaning up to an adult microbiome, which is characterized by this plot here of beta diversity, which measures the relatedness between samples and metagenomic analyses. And this is a plot looking at individual time points of an infant from birth up until weaning. I think this plot ends about 1.5 years compared to the microbiome of its mother. And as you can see, when the infant is on a solely milk-based diet, its microbiome is very distinct to that of its mother. But as the transition from milk to solid food begins, you start to see the slow progression to a microbiome that is primarily being driven by this dietary shift to one that resembles that of an adult. And of course, mice, which is our model species for many of the things that we do, we see a similar phenomenon occur. Mice, of course, develop much more rapidly than we do. And mice typically continue to be on a solely milk-based diet probably between 14 to 16 days old. And then they slowly start to transition in between a mixture of milk and solid food up until 21 days, where we physically wean them from their mothers, and which then they proceed to be on a solely solid food-based diet. And these shifts, similar in humans, are realized in metagenomic data, looking at alpha diversity at day 14. You see a microbiome dominated by mostly facultative anaerobes, which is consistent with the microbiome of a pre-weaning infant. And as the transition to solid food begins, you see the increase in Bacteroides and Clostridial species that are characteristic of an adult microbiome. And this is also visualized in the beta diversity, where you see clustering between day 14 samples and day 18 samples, with day 70 samples on the other end of the axis. And with day 18 samples being split between the two populations, indicating whether these individuals had started eating solid food earlier than others. So the reason why I'm particularly interested in the pediatric microbiome is that several studies have found that perturbations in the early life microbiome has serious impacts on long-term health. And this is in spite of the microbiome returning to what appears to be a normal state after childhood. And this perturbation, generally the studies have attributed this to antibiotic exposure early in life, has been linked to immunological diseases such as asthma and atopic dermatitis specifically. And in particular for my lab studies type 1 diabetes, and my PI, while he was a postdoc, discovered something very interesting in the NOD mouse model of type 1 diabetes, and that when he treats NOD mice with E-alpha-16 NOD mice, which have a E-alpha transgene that protects them from spontaneous diabetes development, when they treat either the pregnant dams or pups 3 weeks that are younger than 3 weeks of age, insulitis score significantly increases, which insulitis is a metric that we use as a precursor to type 1 diabetes development. And likewise, you see an increase in insulitis from 3 to 6 weeks of age, but no difference if vancomycin was given to E-alpha-16 mice post 6 weeks of age. So it indicates that there seems to be an important critical window of protection that involves the pre-weaning microbiome and NOD mice and in the type 1 diabetes model. And I was tasked with investigating the role of the microbiome in protecting from type 1 diabetes, particularly the pre-weaning microbiome. And typically when we want to figure out what a bacteria is doing, the microbiome is, even in a pre-weaning microbiome, is quite complex, and it's difficult to tease apart which members are doing what. So scientists who study these questions typically develop tools using germ-free and gnotobiotic mice for studying host commensal interactions that allow us to whittle down the microbiome to just a handful of species that we are able to either genetically manipulate or track to really get a deeper understanding of the processes involved in this protection. So of course, because this is a commonly used technique in studying these types of interactions, there are gnotobiotic mouse models available and that are well characterized. The most notable would be altered Shadler's flora, or ASF, and a newly developed consortium called Oligo MM12. But neither of these microbiomes were developed to specifically mimic the function and capacity of the pediatric microbiome, which we've determined to be the critical point. So my goal is to ultimately determine how does the early life microbiome impact the long-term health of the host. And to do this, I want to develop a tractable gnotobiotic mouse model of the pediatric intestinal microbiome with the hypothesis that a simplified community of bacteria derived from pre-weaning mice would be able to recapitulate the features of the early life microbiome. So this is just a workflow of how I constructed my pediatric defined community, which we have dubbed PedsCom for short. So here, so we took 14-day-old SPF nod mice from our regular mouse colony, homogenized the small intestine, cecum, and large intestine, and plated those on 4 different media in 3 different conditions: anaerobic, aerobic, and a 5% CO2 enriched environment, and started isolating species to build our culture collection while simultaneously sequencing the cultural plates and the total community to see what type of coverage we could get from culturing, and then selecting what we decided to be at least under 10, and we decided on 9 microbes based on their relative abundance in the donor mice, taxonomic diversity, and as well as KEGG ortholog coverage to try to recapitulate as much function as we could in a handful of species. And after this criteria, we settled on 9 bacteria that we dubbed our PedsCom consortia, which we then inoculated in germ-free Black 6 mice. So this is just looking at the taxa bar plots of our donor mice to get an example of what the microbiome looks like at this age point in our colony. As you can see before, there's a much lower diversity at this time point. You can see in the small intestine, it's dominated primarily by Lactobacilliaceae, as you would probably expect from a mouse that's solely on a milk-based diet. And as you go down the lower GI to the cecum and large intestine, you start to see the introduction of Bacteroides species, particularly Porphyromundiaceae or Tannerelliaceae, which I think is referred to now, a Parabacteroides species, which starts to take up some of this niche. Because of this, these are the 9 members that we've decided upon. So we have Lactobacillus johnsonii, Parabacteroides digitoneus, and Lactobacillus murinus. Those are the top 3 species we saw, and rounded it out taxonomically with an Enterococcus species, 2 Staphylococcus species, 2 Clostridial species, Intestinale and Anorhosipes KK, as well as a Proteobacteria representative, an Enterobacter. Here, this is looking at the read coverage of PedsCom and the total compared to in read depth versus the total community and just the PedsCom members. And you can see that just those 9 members cover from 80% in the small intestine all the way up to 99% of the total read depth in the donor organ sites, while simultaneously only representing around 10% of the microbes present in our adult population, our colony. So we think these microbes are somewhat more related towards juvenile mice than adults. So with our consortia in play, we wanted to answer some questions. Primarily, does PedsCom colonize the mouse, of course, under germ-free settings? Is colonization stable and can it be vertically transmitted? Does weaning impact the relative proportions of PedsCom members? And does PedsCom support the development of an adult immune system? So to do this, we developed 2 gnotobiotic systems and our PBS germ-free controls, where 4 to 6 week old dams are gavaged with our PedsCom consortium and the cecal contents of a 6 week old nod mouse to generate a positive control that we dub SPF-COM, and we allowed these to breed out under isolator conditions and performed our experiments on the F1+ generations. So this is just looking at the orally gavaged, I guess, progenitor mice for our two gnotobiotic communities. Looking at PedsCom, you can see at day 1, what you would expect is dominated by facultative anaerobes, particularly this Enterobacter species. And then just after day 2, Parabacteroides starts to take up more space in the stool, and it stays relatively stable out until 4 weeks. In contrast, you look at the SPF-COM, which exhibits a transition that's more what you would expect in a wild-type mouse during ontogeny, where the first 3 days of post-gavage, you see again a domination from facultative anaerobes, this Enterobacteriaceae, which then starts to make way at 4 days to Clostridial and Bacteroidetes species that are more characteristic of mouse stool. So this transition in staticness in PedsCom is capitulated in the F1+ generations post-weaning. You can see in the SPF-COM a similar transition that seems to be driven by the introduction of solid food, where you start to lose Enterobacteriaceae and Lactobacillus species, and the introduction of S24/7 and Clostridial taxa. While looking at the PedsCom, the relative abundance of the 9 members stays relatively static, even upon introduction of solid food. This is particularly highlighted by looking at PedsCom members in our SPF-COM, where at day 14, they encompass about 50% of the microbiome and slowly whittle down to about an average of less than 5 by 28 days of age. Just to kind of hammer the point home, this is looking again at a beta diversity plot that I had shown previously, where you see a nice clean transition in our SPF-COM mice from day 14 samples to day 21 to finally day 28, and nice clear delineation between those groups. But when you compare to the SPF-COM, not only is there no clear separation between the different ages, they also cluster more similarly to the day 14 SPF-COM sample points. And next, we wanted to, of course, because we study immunology and the host commensal interactions, we wanted to try to use a metric that indicates what would be a wild type transition, an immunological transition from weaning, a weaning marker, which is what I meant to say, a microbially induced weaning marker. And for this, we decided to use the generation of peripheral Tregs that are highlighted by RO gamma positive Helios negative Tregs. And we see in SPF-COM as well as PedsCom an increase in these peripheral Tregs that through weaning, and none in germ-free in the small intestine, as well as in the large intestine, and actually a significantly higher amount of peripheral Tregs in SPF-COM versus PedsCom. And next, we wanted to do a couple of experiments to check the malleability of our PedsCom model. We wanted to introduce a commensal to see if we could indicate any type of immune response to the introduction of a novel species. For that, we used Akkermansia mucinophila, and we introduced this successfully in our PedsCom adults, getting up to very high counts in relative abundance relative to PedsCom. And we also, at least in the cecum and small intestine, saw significant increases in those peripheral Tregs due to the Akkermansia induction. And next, we wanted to see if PedsCom, pediatric mice tend to be more susceptible to salmonella infection, and we wanted to see if PedsCom adults followed that similar phenotype. So we infected SPF-COM and PedsCom adults with salmonella, and we saw a significant difference in mortality between our two gnotobiotic groups, and as well as significant increases in bacterial load in the spleen, pyre patches, and stool. I seem to be running out of time, so I'll skip this because it's preliminary data and just go to my conclusions. So conclusions are, we successfully developed an 8-member pediatric defined community called PedsCom. And we determined that to determine what components of the immune system are dependent on the development of an adult microbiome. We see that PedsCom distribution is stable during the weaning period. PedsCom exhibits stunted gut immunity, leading to lower inducible Treg levels, and PedsCom is more susceptible to salmonella infection. Ongoing work is we're looking at metabolomics and transcriptomic analysis of PedsCom and comparing it to pre-weaning mouse and human microbiomes. Antibody response to PedsCom versus SPF-Com, intestinal histology, looking for development as well as spatial configuration of PedsCom members, and function of PedsCom in our NOD mouse model type 1 diabetes. So with that, I'd like to thank my lab, my PIs, Michael Silverman, and my co-PI, Paul Planet, and all of you for listening. Thank you.
Great, thank you. Looks like we have a question in the chat. And so the question is there a difference in microbiome in breastfed versus infant formula fed newborns?
There is. I only know that literature in passing, but I do know that several studies have found some differences in the microbiomes between the two. That I think that's actually pretty well established, but I can't tell you specifically what those differences are. Yeah.
Great, thank you. So the next question is, are PedsCom mice more susceptible to asthma or other diseases besides pathogen infections?
Well, we haven't really investigated that. I could tell you at least anecdotally, PedsCom mice appear to be healthy. They don't seem to suffer from any health defects in adulthood. There seems to be a little failure to thrive during development that I had to breeze past, but that's still in its very preliminary stages, so I hesitate to say anything about that data. But there might be some early developmental defects, but I don't, at least anecdotally, notice any differences in the adults in terms of health.
Thank you. So we have a little bit of time before our break at 10:05. So if there's any additional questions, you can put them in the chat.
All right, thank you.
Thank you.
Thank you so much, Matt, and thank you so much, Dr. Lubin, for your talk. My name is Michelle Kutzler. I am the student chapter faculty advisor. I'd just like to thank everyone for attending our morning session. Right now we have a break, and I just want to just make a couple announcements. The first is, just share these slides here. Again, we're on break right now, so if everyone could come back for our rest of our morning session, we're going to begin again at 10:15 a.m. with our graduate students and our postdoctoral presentations. And I also want to just invite everyone to stay and enjoy our poster presentations. We have 22 posters that will be presented between odd number posters will be presented between 11:15 and 12:15. And then our even number posters will be presented from 12:15 to 1:15. So during our lunch break today, if you could please stop and visit some of our posters. What I'll do is, this is in the conference booklet that was emailed to all of our registrants, but I will also post this in the chat. And to enter their poster rooms, you click on the Zoom link, and the presenters will be there with their posters. And so I highly encourage everyone to visit our posters at lunch today. And then this afternoon, I would like to invite everyone to stay the whole day. Not only will we be giving out our poster awards and our presentation awards today at the end of our day, but I'd also like to congratulate Dr. Stanley Plotkin, who is receiving the 2020 Sydney PedsCom lectureship. So please stay and return to see our talk later, his talk later today. And again, thanks to our sponsor, PBL-Assay Science, for sponsoring that lectureship. So, all right, thank you so much, everyone. We're going to take a break, and we'll see everyone back at 10:15.
Hello, everyone. Welcome back. I'm going to kick off the next portion of our day. I just wanted to introduce myself once again. My name is Kayla Sakaris. I am from Drexel University. I am the co-VP of the EPA-ASM Student Board, and I work with Dr. Garth Ehrlich. So just a quick reminder, please post all of your questions within the chat. And I have the pleasure of introducing our first graduate student speaker. So first off, the graduate student speaker that will be presenting for this session is Rina Matsuda. They're from University of Pennsylvania, and they're working with Dr. Igor Brodsky. Today, they're going to be presenting their talk titled Inflammatory Monocytes Enhanced Control of Yersinia pseudotuberculosis by Intestinal Pyogranulomas. Rina, welcome, and I look forward to hearing your talk.
Thank you so much for that introduction. So I'll go ahead and share my slides. All right. So hello, everyone. Thank you for coming. I'm Rina Matsuda, and today I'll be talking about how Yersinia infection is controlled in the intestine, following up to two great talks we've had this morning about intestinal infection. And so kind of big picture, the thing that really fascinates me about the study of infectious disease is how over time pathogens and their hosts have really engaged in an evolutionary arms race. And so over time, as pathogens have grown increasingly able to evade the immune response, the host in turn has needed to evolve more complex immune responses in order to deal with these hard to clear pathogens. And so one such example of a complex immune response is granuloma formation. And granulomas are an aggregation of immune cells that are thought to sequester pathogens and prevent their spread. And so granulomas are comprised of a lot of different types of immune cells, which can include macrophages and also other cells like neutrophils that are thought to surround pathogens and really encapsulate them. And so granulomas are a hallmark of both infectious and non-infectious chronic immune stimulation. The most well-studied example are granulomas during tuberculosis infection that form in the lung, but also during parasite infections and non-infectious contexts such as Crohn's disease in the intestine and around this suture in the skin can also induce granuloma formation. But in the context of infection, it's still unclear whether or not granulomas play a protective or pathological role in the control of pathogens. And so in tuberculosis, even though human patients form granulomas that control infection, it's also known that the bacteria can take advantage of this niche in order to replicate and use nutrients there. And so one model pathogen that my lab uses to study the interaction between Yersinia and innate immunity is Yersinia pseudotuberculosis, which is a cousin of Yersinia pestis that causes plague, but Yersinia pseudotuberculosis is an oral pathogen of both humans and mice that first infects the small intestine and then can disseminate to systemic organs, including the spleen, liver, and lungs. And Yersinia is able to block innate immune cell function by use of a type 3 secretion system, which is a needle-like apparatus that injects host cell cytosol directly with bacterial effector proteins known as YOPs in Yersinia. And these YOPs can then block functions of innate immune cells, including phagocytosis and inflammation. And so during Yersinia infection, it's known that granulomas form in draining lymph nodes and peripheral organs. And this has been relatively well characterized in an example here of a mouse lymph node during infection from one of our previous papers shows this round structure that forms that surrounds the bacteria inside of the mesenteric lymph node. And also others have shown in peripheral organs such as the spleen in this immunofluorescence image that Yersinia that's stained in blue in the middle is surrounded by this highly organized collection of immune cells that are recruited that then sequester the bacteria. So here neutrophils are stained in red, and then INOS positive cells that are likely monocytes or macrophages are stained in green that take up distinct spatial spaces around the bacteria. However, although all of this work has been done to characterize granuloma formation in the peripheral organs, relatively little is known about the infection of Yersinia in the intestine. And we think that since this is the first organ that's infected, the gut plays a very key role in functioning as a barrier against Yersinia infection to establish the infection. And so in studying the enteric response to Yersinia during infection, we noticed in the mouse model that when you take a mouse intestine, and what I'm showing here is the intestine that's been cut open and the luminal contents flushed out. When you look at the uninfected control versus the Yersinia infected gut, you can see these macroscopically visible white dots, which are granulomas that form in the intestinal mucosa during Yersinia infection. And to our knowledge, this is the first time in the literature that granuloma formation has been described in the intestine during Yersinia infection in mice. And so when you look at where these granulomas are forming, they are enriched in the jejunum and ileum, which are the distal segments of the small intestine, but also are found in the duodenum and the large intestine. And per mouse, about 20 to 30 of these granulomas form that are visible macroscopically by eye. And so using this system, we're interested in interrogating what factors control granuloma formation and function in the intestine during Yersinia infection. The first question that we wanted to ask is, what are the factors that induce intestinal granuloma formation? And so since Yersinia is an orally acquired bug, it starts out in the lumen and then somehow translocates into the underlying lamina propria underneath the epithelial cells, where it then is engulfed within this granuloma structure. And we want to know what are the bacterial factors that lead to the recruitment and aggregation of these immune cells, and then also what are the host factors that drive the recruitment of these cells from circulation. And next, we want to know how do the immune cells that are recruited then communicate with each other within the granuloma to mediate bacterial restriction and host protection. And so once the cells are here, how do they talk to each other to prevent bacterial escape from the granuloma into the non-granuloma areas of the gut? And then how does that impact downstream spread of the bacteria to systemic organs? So first, we wanted to take a closer look histologically at these granulomas that are forming in the gut. And so what I'm showing you here are small intestines of Yersinia infected or uninfected controls that have been rolled and sectioned. And the granulomas look like this, where there's a large aggregation of immune cells that's underneath the epithelial cells that are organized in villi and crypts up here. And then we also noted that around the granuloma, there's also a lot of epithelial cell hyperplasia or proliferation, which is seen by this bubbling of the epithelial cells that are proliferating. And also we saw that the muscularis, which is the lightly staining area underlying the epithelial cells, is also inflamed and elongated. However, interestingly, when we look at the infected portions of the tissue or the non-granuloma portions of the infected intestine, it looks pretty similar to the uninfected controls, suggesting that Yersinia infection in the intestine is highly focal and localized to the granulomas. Next, we were curious if these granulomas contain viable bacteria. And so we took transverse sections of the small intestine, and when you look at the granuloma itself and you zoom in, we noticed that this white arrow is pointing to small microcolonies of bacteria that stain in a more light color than the kind of sea of nuclei that surround it that stain more darkly, that are the immune cells. And then next, we took advantage of the fact that we could macroscopically identify these structures And we used a punch biopsy tool in order to biopsy the granulomas, as well as the non-granuloma areas of the infected intestine. And when we plate these on agar, we find that the granulomas have a very high burden of live Yersinia, whereas the non-granuloma areas have a low burden, again suggesting that Yersinia infection in the intestine is highly focal and concentrated to the granulomas. So next, we wanted to investigate the role of bacterial virulence and bacterial factors in the formation of intestinal granulomas during Yersinia infection. And so, as I talked about before, Yersinia uses a type 3 secretion system to inject YOPs that then block innate immune cell function. And Yersinia is a very genetically tractable bacteria, especially because its type 3 secretion system and all of the YOP effector proteins are encoded on one virulence plasmid that we can genetically ablate. And so when we take this plasmid cured strain that no longer expresses its type 3 secretion system in the dark gray circles, and we infect these bacteria into mice, we find that in the pyrus patches and mesenteric lymph nodes, which are the lymphoid tissues of the intestine, the plasmid cured bacteria are able to colonize the intestinal lymphoid tissue, although maybe to a lower degree in the pyrus patches, which is consistent with the published literature. But when we look at granuloma formation by eye, we find that we cannot detect any granulomas in the plasmid cured infected mice, showing that the type 3 secretion system is necessary for granuloma formation during Yersinia infection. And next, to bring you to a model slide, we've shown that the Yersinia type 3 secretion system is necessary for the granuloma formation, but we don't yet know what cells are being targeted and what cues are being released that then lead to secretion system dependent granuloma formation in the intestine. So next, we wanted to start to characterize the host response side of things in granuloma formation. And we wanted to start to take a look at what types of cells were in these granulomas. And to do this, we wanted to take a flow cytometry approach. So first, we hypothesized that monocytes and macrophages would be present in these structures, since these cells are very common in other granulomas in infection. And so these cells can be distinguished by a surface marker CD64. Next, we hypothesized that neutrophils would also be present based on the histology, and these cells by flow can be characterized by the surface marker Ly6G. So first, to start with the monocytes and macrophages, again, which are CD64 positive, when we took out the granulomas from the mice, and then we looked at the surface marker CD64 on all of the viable immune cells, we found this population of cells that was CD64 positive. And then next, when we compared the granulomas to non-granuloma infected tissue and uninfected controls, and then we looked at the CD64 positive population of monocytes and macrophages, we can identify different subsets within this population by looking at the expression of Ly6C and MHC class II. And so when we take the Ly6C high cells as monocytes and then the Ly6C low cells as macrophages, and we quantify those populations between these three different samples, you can see that the granulomas specifically are highly enriched in both monocytes and macrophages compared to the non-granuloma tissue and the uninfected control tissue. And then next, when we looked at Ly6G, which is the surface marker for neutrophils, there's a lot of neutrophils as well in these granulomas. And so next, we wanted to ask functional questions about the role of monocytes in these Yersinia granulomas. And so we took CCR2 deficient mice, which are kind of deficient in monocytes since CCR2 is necessary for monocyte egress out of the bone marrow. And these mice are denoted as GFP since GFP is knocked into the locus. And so the homozygous mice are deficient in CCR2. And when we did this and we looked by eye, the granulomas still form. However, when we looked at the bacterial burdens, while we found no difference within the granulomas, we found that in the non-granuloma areas of the intestine, the monocyte deficient mice have a higher bacterial burden, suggesting that Yersinia control is defective in the non-granuloma areas of the intestine of these mice. And when we looked histologically at the structure of the granulomas in these mice, we found that in the CCR2 monocyte deficient mice, the bacteria look cloud-like and larger compared to the small bacterial microcolonies and the wild-type granulomas with the white arrows. And then also there seems to be fewer neutrophils, which are denoted by this black arrow around the granuloma. And when we have blinded scoring done by pathologists, although the overall inflammation is not different between the two groups, when we look specifically at the number of bacterial colonies and also specifically at the ones without inflammation, we find that while in the wild-type setting, all the bacteria are surrounded by inflammation, in the absence of monocytes, there are these colonies that don't have inflammation. And so to bring you to a model slide, in the wild-type setting, granuloma formation happens and bacterial controlled in the intestine. However, in the CCR2 knockout background, there seems to be an architectural defect in the granulomas where there's a central area of necrosis that happens and bacterial escape. And so to go a little bit faster, since I'm running out of time, in the monocyte deficient mice, the bacterial burdens are higher in a lot of the peripheral organs tested, and these mice also succumb to acute infection. And so to bring back to the model, in the monocyte deficient mice, there's a defect in granulomas and also a defect in the systemic organ control of Yersinia. And so to wrap up, we've found that Yersinia pseudotuberculosis is providing a tractable model to study granuloma formation in the intestine during bacterial infection. And so we have future directions to interrogate the roles of inflammatory monocytes as well as neutrophils in the control of Yersinia in the intestine. And so I'd like to acknowledge my lab, especially Daniel, who's pictured here, who I work closely with in order to do this project. And also the people at the Comparative Pathology Core who've been really instrumental in the histological assessment of these granulomas. And with that, I'll be happy to take any questions.
Thank you so much, Rena. Wonderful talk. There are some questions in the chat. The first is from Dr. Kutzler, and she asked, what types of pro-inflammatory cytokines are macrophages secreting? Is NCP1 chemokine also important in mediating the function of the macrophages?
Right, yeah, so that's a really good question. So we've profiled some cytokine production in these granulomas, and we know that the pretty common innate cytokines such as like IL-1 beta, IL-6, TNF are produced in the granulomas. And we also know that there's a role for TNF in the actual control of bacteria within the granulomas. And then you specifically asked about MCP1, which is CCL2, which acts directly on CCR2 that I was talking about. And so we know that in the CCR2 knockout mice, there's a lot of MCP1 that accumulates probably since it's not binding to its receptor. But we're not sure if there's a role locally in the intestine for MCP1, or if it's just kind of a facet of the model where the receptor is knocked out and then the monocytes get stuck in the bone marrow.
Perfect. And I think we have time for one more. The next question is, are wild-type mice susceptible to Yersinia pseudotuberculosis infection in this model? Meaning, do they clear the bacteria, or is it background dependent, mainly B6 versus BLBC versus 129? Yeah,
that's a great question. So in the wild-type setting, it is strain dependent, and then it's known that the C57 Black 6 mice that we use are resistant to infection, and they clear the infection, after a few weeks. Whereas I think BALB-C mice are more susceptible, and tend to succumb to infection. I'm not sure if it's 100%, but I think they're more susceptible than the black 6 mice.
Thank you. And unfortunately, Rena, that's all the time we have. There are a couple of questions which I will send to you shortly. Thank you so much. And I believe the next person is up.
Sorry, you didn't hear me before. Thanks, Kayla. Good morning, everybody. My name's Ron Lucarelli. I'm a graduate student at Temple University, and I'm the current secretary on the Student Leadership Board. And this morning, it's my honor to introduce our next student speaker, Alicia Segrist. She's in the lab of Dr. Sarah Cherry at the University of Pennsylvania, and she's going to be giving a talk entitled Microbiota-Derived Cyclic Dinucleotides Drive D-Sting Dependent enteric antiviral immunity in enterocytes. Alicia, when you're ready, the room is yours.
Okay, thank you for that introduction. I just also would like to say, like, I'm usually the only person talking about Drosophila, so it was really great to be able to hear Dr. Watnick's talk this morning. Okay, so, sorry, it's not letting me advance my slides. Okay, so as everyone at this talk knows, the microbiota has been described as influencing many different aspects of intestinal biology and immunity. This cartoon here just shows how some of these that have been described, like short-chain fatty acids, either interact with the epithelium or with the innate immune cells in the lamina propria. And while some of these compounds and how they interact with the epithelium and the immune cells have been characterized, there are still many other microbial mediators whose influence on immunity is unknown. So to address this, the Cherry Lab uses Drosophila as a model because of the powerful genetic tools like the UAS-Gal4 system, which allows us to specifically overexpress or knock down a gene of interest in the whole animal or in a cell and tissue specific manner. And since we're interested in studying the microbiome, Drosophila has a much more simple microbiome. There are 5 major species that make up the majority of the microbiome, with both gram-negative and gram-positive bacteria present. And one of the species, Lactobacillus plantarum, is also found in the human microbiome. So the simplicity makes it much easier to understand functionally how each species is interacting with the intestine. And there's functional and cellular conservation of the intestine. So this cartoon on the right has the areas of the mammalian intestine and Drosophila intestine that perform the same function color-coded. And there are the same cell types like enterocytes, enteroendocrine cells, and intestinal stem cells, and similar signaling pathways that control differentiation and proliferation of the intestine. Since I'm interested in the role of the microbiome during infection, I just started with looking to see how the microbiome influences either oral infection or systemic infection. So this cartoon on the left on the top is a diagram of how oral infections are done. So I take flies and either mock or antibiotic treat them. So antibiotics are directly added on top of the food, so the flies are forced to eat it. They're starved for an hour and then placed on vials that has the virus, Symbis virus, which is an alpha virus that I'll be using throughout the rest of the talk. And then at 6 or 7 days post-infection, I dissect the intestines and then pool 15 intestines together and then do qPCR to quantify viral RNA levels. Basically the same thing is repeated, but instead of starving them and orally infecting them, the flies are injected with virus, and then at 7 days they're collected for qPCR. So during oral infection, antibiotic treatment results in a significant increase in Symbis infection of the intestine. But during systemic infection, antibiotic treatment doesn't influence the amount of Symbiose RNA in flies. And since the data I just showed you suggests that the microbiota is providing some compounds or some interaction with the intestine that is resulting in protection from infection, we're interested in characterizing what these compounds could be. And one of the things we're interested in are cyclic dinucleotides, which are these essential secondary messengers in bacteria that are basically produced by all bacteria that either make C-di-AMP or C-di-GMP. So to see if it's likely that these compounds are produced by the 5 major commensals that we find in the microbiome of the Drosophila we have in our lab, I just looked at the sequences deposited in CBI to see if any of them contain a GGDEF domain, which is the domain present in all diguanylate cyclase enzymes, which are responsible for producing C-di-GMP. And so 4 of the 5 commensals do contain this protein, suggesting they are producing cyclic dinucleotides. So we're interested in cyclic dinucleotides because of their known role in interaction with STING, which is this broadly antimicrobial protein that is antiviral against DNA viruses and against bacteria. So STING is activated by binding cyclic dinucleotides, and these can come from bacteria or from the endogenous DNA binder cGAS. And since it seems that the Drosophila microbiome is producing these cyclic dinucleotides, we hypothesize that the microbiome might be providing this pool of cyclic dinucleotides that are then interacting with STING to inhibit RNA virus infection. So the next experiment I wanted to do is just to see if STING is antiviral in the intestine. So I either orally infected flies or systemically infected flies. And during oral infection, I saw STING is antiviral, but it's not antiviral during systemic infection. So as I mentioned previously, we can specifically knock STING down in different cell types. So I either knocked STING down only in enterocytes, only in intestinal stem cells, or only in enteroendocrine cells. So when STING is only knocked down in enterocytes, the intestinal stem cells and enteroendocrine cells are still producing STING. And I see STING is specifically antiviral in enterocytes. Since I saw STING as antiviral and I know the microbiome is protective, the next experiment I wanted to do was to see if cyclic dinucleotides influenced virus infection at all. So I took flies, antibiotic treated them, and then either fed Symbiose or fed Symbiose and C-di-GMP at the same time. So during oral infection, I see that antibiotic treatment results in a significant increase in infection, and that feeding of C-di-GMP to antibiotic-treated flies is sufficient to protect from this increase in infection seen by loss of the microbiota. However, when flies that are either conventional, which is the data I'm showing here, so they have a microbiome, or flies are antibiotic treated, which data I'm not showing, cyclic dinucleotides are not antiviral during systemic Symbiose infection. Since I see during oral infection that C-di-GMP is protective, I next wanted to know whether this is STING dependent. So I set up a very similar experiment, but I used either control flies or flies where STING is knocked down only in enterocytes. And I see in these driver control flies that C-di-GMP is still protective, but this protection is lost when STING is absent in enterocytes, indicating that STING is required for C-di-GMP mediated protection from infection. So in mammalian biology, STING is, once activated, is described as acting through interferon and NF-kappa B signaling and other less characterized pathways like autophagy activation. And it typically interacts with these downstream effector pathways through the protein TBK1. So Drosophila only has this gene called IK2, which is highly similar to human TBK1 and IKK epsilon. And I'll be referring to as TBK1 in the rest of the talk. But it hasn't had a well-characterized role in antiviral immunity in Drosophila before. So I took flies where TBK1 is knocked down in enterocytes and infected with Symbis, and I see that TBK1 is antiviral, but TBK1 is not antiviral during systemic infection. And I next wanted to know if TBK1 is downstream of sting in Drosophila, then I would expect it to be required for CDI-GMP's protection from infection. So I fed either control flies or flies where TBK1 is knocked down in enterocytes, and I see that CDI-GMP protection is lost when TBK1 is knocked down. So after finding that cyclic dinucleotides are antiviral and that this antiviral activity requires sting in TBK1, the next thing I wanted to address is what other downstream pathways is sting acting through to inhibit virus infection. So as I mentioned, mammalian sting acts through interferon, NF-kappa B, and other pathways. Drosophila don't have interferon signaling, so I wasn't able to look at that, but they do have NF-kappa B, and unfortunately I don't have time to show you that data, but in the intestine at least, it looks like sting and NF-kappa B are not involved in the same pathway. So I next decided to look at the role of autophagy in Symbis infection. So I just inspected a panel of flies where essential autophagy genes were knocked down. And I see when ATG5, P62, which is a cargo receptor for autophagy, and when ATG16 are knocked down, there's an increase in Symbis RNA levels in the intestine, suggesting that autophagy is antiviral in the intestine. So I next wanted to see what is activating autophagy and whether infection of Symbis is what is inducing autophagy in the intestine. So when autophagy is activated, a cargo receptor like P62, which I just showed you is antiviral, binds a ubiquitinated cargo, like a viral protein, and then recruits autophagy proteins and membranes, and then this surrounds the cargo. So on this membrane, the autophagy protein ATG8 is conjugated to the membrane. So there are these flies where ATG8 is tagged with MCherry. So you can visualize autophagy occurring through ATG8 puncta formation by immunofluorescence. So I took these flies and either fed rapamycin, which is an activator of autophagy, which should increase the number of ATG8 puncta that I see in the intestine, which is indeed what I see, or I fed Symbis virus. And I see that Symbis virus also increases the number of ATG8 puncta in the intestine. So that leads me to this model where the microbiota is potentially providing this pool of cyclic dinucleotides, and these cyclic dinucleotides block Symbis infection in a sting and TBK1 dependent manner. I also see that autophagy is antiviral in the intestine, but I need to do more work to determine whether autophagy is induced by Symbis or whether feeding of cyclic dinucleotides themselves can influence autophagy induction, and then also determine if this is sting and TBK1 dependent. I've also shown that there are clearly tissue differences in how the microbiome influences Symbis infection. So I'd like to do more work to characterize what these differences are. And then there's a question of how cyclic dinucleotides are entering cells, and because most in vitro experiments require transfection of cyclic dinucleotides into cells, and in the last year or two, there have been an increasing number of reports about cyclic dinucleotide transporters. So it's also something I'm interested in investigating further. And I'd like to thank everyone in the Cherry Lab. I should point out that Beth Gould is a technician in the lab that did all of the injections for the systemic infections that I showed you guys. I'd like to thank my thesis committee for their input and funding sources, and I'm happy to take any questions now that you might have.
Great talk, Alicia. I did not see, oh, wait, I think one just came in. So you have a question. Cyclic di-GMP is a bacteria cytoplasmic second messenger. Do you think bacteria need to die for release of the cyclic di-GMP, or is there an exporter?
Yeah, that's a really good question. As far as I know, C-di-AMP and bacteria have known exporters, but for C-di-GMP, there hasn't been an equivalent exporter found. So it is possible that they do have an exporter and it just hasn't been discovered yet. And I also think it's likely that the microbiota are dying. As they're dying, they're releasing this. So it could potentially, both could be playing a role in providing the cyclic dinucleotides in the intestine.
Okay.
And it's unclear at the moment which it is.
Next question for you is this. Which groups of microbiota would be depleted by your antibiotic treatment? Do you know which specific bacteria groups would provide this antiviral protection?
Yeah, so when we antibiotic treat, I didn't show, I guess, the plating data just because it kind of clutters up the slide, but all of the species are cleared. So both gram-negative and gram-positive are gone. And from the sequences that I looked at in NCBI, both gram-negatives and gram-positives produce C-di-GMP. And gram-positives also usually produce C-di-AMP. So it seems like there's probably a role for both in this signaling.
Very good. One more question. Do pathogenic bacteria translocate higher levels of CDI nucleotide and thus induce higher levels of antiviral response? At least in Listeria, the multi-drug efflux pump can secrete CDI-AMP. That might have been a mouthful for you, sorry.
So I've never done an experiment in which I infected flies with bacteria that I know, like Listeria, that would be producing CDI-AMP and then also infected. I do have Listeria and E. coli strains that, in the case of Listeria, lack the export pump, so then I know there would be no CDI-AMP production, or E. coli where they're overexpressing CDI-AMP. And because I think that's a great experiment to do if I can see correlation of more or less CDI-AMP with protection or not from infection. There's just been a technical issue with trying to get the bacteria to colonize the intestine, which has been pretty frustrating because I think that would be a great thing to do. Just technically it hasn't worked out.
Very good. Thank you so much for speaking today. We have one more minute, but you answered all your questions. So if you have any further, you could message Alicia directly on the chat function. I'm going to pass it over to Marissa to introduce our next student speaker.
Hi, everyone. My name is Marissa Egan, and I am a third-year graduate student at the University of Pennsylvania. I'm currently the treasurer of the EPA-ASM Student Chapter Board. Our next speaker is Dr. Ruchika Dehanwal, a postdoctoral fellow from Dr. Dieter Schifferli's lab at the University of Pennsylvania. She will be presenting her really exciting and impressive work demonstrating that Salmonella OMVs serve as traps for antibacterial host molecules. Take it away, Ruchika.
Hi, everyone. I hope everyone can hear me. Let me just share my screen. There you go. I hope this is visible.
Yep, we can see it. Thank you.
So today I would be talking about bacterial outer membrane vesicles and how they interact with host antimicrobial components and how this helps in bacterial pathogenesis. The pathogen of interest in our lab is Salmonella enterica. Okay, there we go. Is Salmonella enterica, which is a gram-negative intracellular pathogen. This infects via the oral route, and once into the intestine, it crosses the intestinal epithelial barrier, where it is then taken up by the macrophages. Since Salmonella is an intracellular pathogen, it can both survive and replicate very well within the host cell. When in the host cell, the bacterium is contained in a very specialized vacuole called as Salmonella-containing vacuole, where the host cell tries to employ several stress effectors on the bacteria, trying to kill it. For example, low acidic pH or low nutrient availability and cationic antimicrobial peptides. But Salmonella is a very smart pathogen. It activates its two-component system, 4P4Q, which help in resistance against all these stress effectors and therefore survival of the bacteria. The 4P4Q is an environmental sensor. It senses the low pH and low magnesium in the vacuole, thereby activating a set of genes called as PAG, or the 4PQ activated genes, or represses another set of genes called as PRGs, which is the 4PQ repressed genes. This activation and repression leads to downregulation of SPY1 type 3 secretion system and upregulation of SPY2 type 3 secretion system, which helps in nutrient acquisition and therefore the bacterial survival. The 4P4Q also leads to an altered antigen presentation on the bacterial surface proteome. It also modifies the outer membrane components of the bacteria like LPS, therefore increasing the defense of the bacteria against cationic antimicrobial peptides. Talking about the bacterial outer membrane, the outer membrane is made up of a phospholipid bilayer decorated with several outer membrane proteins, and with these branch structures called as LPS. LPS, or the lipopolysaccharide, is the major antigenic component of any gram-negative bacteria. This LPS is made up of three regions: an outer O antigen, a middle core, and an inner lipid A. The lipid A of the bacteria at 7.6 pH chemically looks somewhat like this, with two negatively charged phosphate groups, which are highly approachable by the cationic antimicrobial peptide and therefore can kill the bacteria. But when the bacterium is present in the vacuole, it senses the low pH and low magnesium, therefore activating the 4PQ system, which then derivatize these phosphates with phosphoethanolamine, which is a more positively charged moiety, therefore making the bacterial outer membrane more resistant to cationic antimicrobial peptides. 4PQ activation also activates other genes like LPXO that hydroxylates lipid A, PAG-P that adds a palmitoyl chain to the lipid A, and PAG-L that deacylates lipid A. These changes make the lipid A and therefore the bacterial outer membrane more stable for survival under the stressed conditions. Recently, it was identified that when a bacteria is grown in 7.6 pH and high magnesium conditions, recapitulating the extracellular environmental conditions, is when transitioned into a low pH and low magnesium condition, recapitulating the intravascular conditions, the bacteria now starts to increase its OMV production. OMV production, or the outer membrane vesicles, are nothing but 20 to 200 nanometer big vesicles that are derived from the outer membrane of the bacteria. These contain periplasm in its lumen, along with some toxins and some proteins. These vesicles help in eliciting inflammatory response, delivery of toxins, and cell-to-cell communication. The group also identified that this increase in OMV production is 4PQ dependent. So we asked, what are the 4PQ activated genes that are involved in OMV production? Also, what is the role of these 4PQ induced OMVs in bacterial pathogenesis? So for our experiments, we grew the bacteria in low pH and low magnesium conditions for activation of 4PQ. And we isolated OMBs from these bacteria and analyzed by transmission electron microscopy and nanoparticle tracking analysis. The nanoparticle tracking analysis is a technique which is based on the principle that when these tiny vesicles are bombarded with laser, they reflect light. And this reflected light is then detected by a detector, and therefore can give us a fair estimation of size and the concentration of the vesicles. So for our experiments, we created several gene deletion mutants of various 4PQ activated genes and analyzed how many OMBs are being produced by this particular bacteria now. Recently, it was identified that PAGP is an important player in OMB production. And also, we saw that deletion of PAGP reduced the number of vesicles being produced by the bacteria and therefore served as a very good positive control. We also identified another novel gene called as PADC, which when deleted, significantly reduced the number of vesicles being produced, as much as the deletion of 4P gene itself. We also analyzed other PAD genes like PAGN, PAGL, PGTE, LPXO, and PMRAB, and found that the deletion of these genes had a very weak effect on the OMB production, suggesting that PADC is an important regulator for OMB production, and therefore we focused on this for our later studies. So we used in our next experiment a 4PC strain. A 4PC strain is a strain where 4P is constitutively active and therefore produces significantly high number of vesicles in comparison to the wild type. And when PADC was deleted in a 4PC background, the number of vesicles produced went significantly down. In fact, when we overexpress PADC in a 4P deletion mutant background, the number of vesicles produced went significantly up, suggesting that PADC is an important regulator for OMB production. Next, we wanted to characterize what is the role of these 4PQ-induced OMBs in bacterial pathogenesis. And so we asked, what is PADC? Now, PADC is an outer membrane protein that has two homologs in Salmonella, namely RCK and OMPX. There's another homolog in Yersinia pestis, which is called as ALE. And this is all that we know about PADC right now. But we also know that RCK and ALE are two very important complement evasion proteins for Salmonella and Yersinia, and both can inhibit classical lectin and alternative pathway of complement. So consequent question arises, can PADC also resist complement attack or not? And so there was a study done in back 1994 by Heffen and et al., where they suggested that PADC is not a complement evasion protein. But a more recent study by Nishio et al. In Salmonella cholerae suggested that PADC is a complement evasion protein. And since there was a contradiction between the two reports, so we asked, is the typhimurium PADC a complement evasion protein or not? To which we answered that yes, PADC from typhimurium does mediate resistance to complement attack. In this experiment, we took bacteria that were grown under 4PQ inducing conditions and incubated it with human serum. After 60 minutes, the survival of the bacteria was calculated. And we found that deletion of PADC, just like the deletion of RCK, which is a known complement evasion protein of Salmonella, made the bacteria highly susceptible to getting killed by complement, just like the deletion of the 4P gene itself, suggesting that PADC does mediate resistance to complement attack. We used a PGTE gene deletion mutant as a positive control and analyzed other PAG mutants also and found that deletion of these other genes like PAGP or PAGL did not mediate any resistance to complement attack, suggesting that PADC is involved in resistance to complement attack. But are the OMVs produced by PADC also important? To analyze this, we used purified OMVs in equal number, both from wild-type bacteria and PADC mutant bacteria, indicated by red and blue over here, and incubated them with the wild-type Salmonella typhimurium in the presence of serum, and found that with increasing number of OMVs incubated with the bacterial survival also increased in a dose-dependent manner. In fact, the survival of the bacteria was more enhanced when the OMVs that were incubated with the bacteria were isolated from 5.8 pH, that is the 4PQ inducing conditions, suggesting that only the PADC expressing OMBs that were isolated under the 4PQ inducing conditions are protective to the bacteria in a dose dependent manner. And we found this profile true for both wild type typhimurium and also for PADC deletion mutant. But are the PADC homologs RCK, OMPX, and ALE also involved in OMB production? To answer this, we found that neither the deletion of RCK, OMPX or overexpression of RCK affected the OMB production. Only the PADC deletion reduced the number of vesicles, and PADC overproduction increased the number of vesicles and Salmonella. Also, the expression of ALE, a Yersinia protein in Salmonella, did not affect the OMB production, suggesting that only PADC and not RCK and OMPX or ALE mediate resistance to complement attack in an OMB dependent manner. Later, we asked what part of the complement. We have to first understand that the complement system can be divided into three: alternative, lectin, and classical pathway, all of which are dependent on the presence of a divalent cation. Where classical and lectin pathway require a calcium ion, alternative pathway requires a magnesium ion. So if you add EGTA to the complex, you can block both classical and lectin pathway. And if you add EDTA, you can block all the three pathways, which we did. We incubated the bacteria with the serum in the presence of EGTA or EDTA, and found that when the bacteria were incubated in the presence of EGTA and the alternative pathway was still active, the bacteria was still getting killed. But when we added EDTA and all three pathways were blocked, the bacteria started to survive more. That means the bacteria are getting killed in an alternative pathway dependent manner. And when we incubated OMBs, and more so the PADC expressing wild type OMBs, the bacteria started to survive even more, suggesting that these bacteria are blocking the alternative pathway of complement. But what component of the complement? Like, there are several key players involved. Since C3 is a very important player in all the three pathways, we focused on C3 first. C3 is a very big protein, which when activated is cleaved into C3A and C3B. C3B then goes and binds to the bacterial surface, activating the membrane attack complex formation and kills the bacteria. But when there are some complement inhibitors present in the complex, like factor H, factor I, or vitronectin, this C3b is degraded into IC3b, or the inactive C3b, which cannot kill the bacteria, and the bacteria survives. And this IC3b can be detected on the gel as a 68 kilodalton and 46 kilodalton band. So for our next experiment, we used equal number of purified OMBs from wild type and PADC mutant bacteria and incubated it with serum. These OMBs were then pelleted, and a supernatant containing unbound complement and the pellets containing complement bound to the OMBs was collected. This was then ran on gel and analyzed for the presence of C3b. And we found that only the wild type OMBs showed the presence of a 68 kilodalton and 46 kilodalton band, and not the PADC mutant OMBs, suggesting that these PADC expressing wild type OMBs not just bound to the C3b, but also were able to degrade it. We also found that incubation of bacterial cells with the OMBs significantly inhibited the C3 deposition on bacterial surface, as we found there was a significantly low amount of C3b associated with the bacteria when they were incubated with PADC expressing OMBs, in comparison to the bacteria that were incubated without any OMBs or bacteria that were incubated with RCK expressing OMBs. Which means these PADC expressing OMBs not just bind to C3, but also degrade it. But how does it do so? Does it recruit some kind of complement inhibitor? And so we incubated equal number of OMBs from wild type PADC mutant and RCK mutant bacteria with the human serum, and found that all three were able to recruit a complement inhibitor called as factor H. PADC mutant could be recruiting factor H due to the presence of RCK and PGTE, both of which are known complement evasion proteins of Salmonella and can recruit factor H. RCK could also be recruiting this factor H due to the presence of PGTE and maybe because of PADC. And therefore, we created a 4 mutant where RCK, OMPX, PADC, and PGTE all were absent. And then overexpressed PADC and RCK in this 4 mutant background and found that expression of PADC made these OMVs recruit a significantly high amount of factor H in comparison to OMVs that expressed RCK. In fact, when we incubated recombinant protein PADC and RCK with purified factor H, both were able to recruit factor H, but PADC did so more strongly, suggesting that these PADC expressing OMVs recruit complement inhibitor factor H that helps in degradation of C3b. Therefore, in the end, we conclude that Salmonella typhimurium resists complement attack by both the expression of PADC and by increasing the OMV production that help in degradation of the C3. We propose that these OMVs act as decoy molecules that help in C3b depletion, therefore preventing the membrane attack complex formation on the bacteria and hence the survival. In the end, I would like to thank the members of Shifreli Lab, Brodsky Lab, and Shin Lab for helping with various experiments and suggestions, Prashant's Lab for helping with nanosite, and Women Lab for helping with the creation of various constructs. I'm open to questions.
Thank you, Ruchika. That was an amazing talk, really beautiful data. Looks like we have a question in the chat from Dr. Tam. He asked, when you induce OMV production, what are the relative abundance of PAG-C in OMV versus bacteria? Are PAG-C differentially trafficked to the OMV?
Just one moment. How do I? Okay. The question was when you induce OMV. Okay, we are testing this. We are doing a proteomic analysis and we want to identify how much protein is present, how much PADC is present in the bacterial outer membrane versus the OMVs. But there was a study which was recently published which suggested that PADC is preferentially enriched in the OMVs. But what's the percentage, we do not know yet, and we do want to analyze that.
Thanks so much. Let's see if other questions pour in. In the meantime, I actually have a curiosity question. So really neat work with Salmonella typhimurium. Has anyone, or are you interested in characterizing OMV production in other reservoirs of Salmonella, like typhi for the human part?
Yes, we have done a PADC deletion mutant in Salmonella typhi also, and in typhi also we found that deletion of PADC does reduce the OMV production. Not as much as the typhimurium, but it does reduce it.
Okay, interesting. Thanks, Ruchika. So if anyone has any other questions, please place them in the chat. Okay, Dr. Kutzler has a question. She said, beautiful data. Since OMV functions to inhibit those immunity, does the function change in biofilms? Does that play a role?
That's a very interesting question. Does the function of OMV change in biofilms? We do not know, but in OMVs do help in cell-to-cell communication, in bacteria also, and they do help in biofilm formation. That's also a study which has already been done. But what components do they interact with for the biofilm formation, we do not know. And that would be a very interesting thing to be studied.
Okay, great. Thank you so much, Ruchika, for an amazing talk. And thank you to all of the speakers for this morning's really great and exciting session. Now I'm going to hand it over to Dr. Kutzler before we have our poster sessions and lunch break. Thanks.
Thank you very much. Again, I'd like to echo Marissa's comments. What a great morning session. Thanks everyone for attending. I'm going to just share a few slides just to give some instructions for the rest of the day. So right now we are ready to start our lunch break. And I just would like to ask that our attendees please attend and visit our poster presentations. We have 2 sessions today. The odd number posters, the presenters will be in their Zoom rooms from 11:15 to 12:15. And then our even number poster presenters will be in their Zoom rooms from 12:15 to 1:15. And in the conference abstract booklet that you received this morning by email, I also dropped it into the chat. So if you scroll up, there is a PDF that looks like this, where we have all of our poster presenters, their names, institutions, their abstract titles. Their abstracts are in the conference booklet, and then there's a link to their presentation room in this document. Some of our Zoom rooms require a password, and that is all the information you need to be able to visit our poster presenters are shown here. And so have lunch, take a break, visit our posters, and then we want to make sure to invite you back for our early afternoon session, where we have Dr. Taji Harris from the University of Virginia School of Medicine leading us off at 1:30 p.m. So after you visit our posters, please return to see Dr. Harris, as well as Ron and Hager, two of our graduate students who were selected to give talks today. And then after the early afternoon session, we have our final session, which includes postdoctoral fellow, Dr. Rosende Mela Silva from Dr. Segal's lab. And then we have our 2020 PESCA lecturer, Dr. Stanley Plotkin, who will finish the day today. So with that, again, I'd like to thank the student officers for moderating the talks this morning, Dr. Vincent Tam for being our guru, helping us with our YouTube live stream and our Zoom today, as well as all of our morning speakers for such a great session. All right, thank you very much, and we'll see you back at 1:30.